So we can look at those. Now let me shift to the next problem, which is the segmentation. Again, to our analogy of the circuit tracing problem, if you had a circuit board with ball pin arrays of lots of holes from the processors, it would be hard to identify where are the edges from one chip to the other. That's rather important if you want to interpret the information flow that's going on in that system. So we need a method of segmenting and finding the borders of the areas, putting them in alignment with the true anatomical and functional borders. So this is an illustration of my brain, and if one looks in, it looks like just a mess of fibers connecting within that system. So to do this, one needs techniques to segment it, and this is a technique we have been using as an illustration. So you start with a connection matrix. That is, of all tens of thousands of locations on the cortical surface, what point is connected to another point, where the color here indicates the strength of connection of fibers within that process. Then you use some method to cluster the matrix of connections within that set into groups so that they are only connected within their cluster. Then you can look within that cluster and for each location identify what is the correlation of each location relative to its neighbor, and you find sharp edges in that drop-off as illustrated by the box. And then you go back to the cortical area and define the edges of the cortical areas based on when you see sharp drop-offs in the correlation of the connective topology. This is one method. We are hoping in the competition that we will spur many groups to try different methods to accomplish this critical problem of cortical segmentation. So let us give you an example of an overlap test that we've done, and it illustrates some of the data for the competition. We can identify the borders of visual areas, particularly V1 and V2, by doing rotating checkerboards. So what we have here is a expanded or inflated view of the occipital pole of this individual, with the colors indicating the ratio of times in 5 separate runs this area was coded as V1, the dark blue area, or V2, the orange area. And I want you to note that the borders represent very sharp borders. That is, within 2 millimeters, we can identify where the edges of these are within an individual. Then we can use techniques to identify those borders based on either the anatomical or the resting state connectivity. This is an example of one of the approaches we have used. And then we can contrast the overlap. In this particular case, we can look at the overlap in an area such as the region in the lower left, where we've slightly missed the division between V1 and V2. And we then get a metric of the degree of match that we have within the areas. Now, recently, there's also been important progress in brain and graph theoretic approaches to brain topology. So they're developing tool sets to create and analyze network topology based on the anatomical and functional links, giving us degree of overlap, paths and distances, subnetworks, motifs within that process, and modularity and structure of those networks. So these recent advances of a substantial literature from the primate and human anatomical and functional data, the ability to follow fibers through multiple tracks, the developing segmentation methods, and this developing theoretical analysis method give us hope that we are at the point for important progress in creating a complete human connectome. So now let me shift to how the competition will work. I'm going to describe the data that will be available and how we will score the entries. Notice that the data availability and scoring methods are subject to change. This is the first time we've ever done a brain connectivity competition. We're still collecting data and putting together the procedures. We have a scientific board, which I will describe at the end, that we evaluate ways of doing the scoring and the data to maximize the potential progress that we might have. Please watch the website for updates and changes within this process. We will send notices to registered members about any changes. Please treat these as the expected or initial set of specifications, but watch the website for updates. So the data for the competition. The data were collected at the University of Pittsburgh on 4 volunteers in a 3T TIM Trio scanner with a 32-channel head coil. This is one of the best combinations of equipment to do this kind of work. It's a very high-resolution, state-of-the-art diffusion, structural, and functional activity data set. The planned data for the competition will include 4 subjects. The first subject we call the development subject. We will give you all the data of both the structural and functional data sets that we will be using within that set. We will also provide 3 competition data sets of 3 subjects where there will be validation data which will not be released until after the scoring is completed. That is, we will use it to score the elements, but we will expect you to find borders for the example without some of that data. The MRI data, there will be structural, diffusion, and functional resting state on all 4 of the subjects. We will also collect fMRI retinotopy, tonotopy, the language network, cognitive control network, and the motor system. The data will be processed and will provide you the benefits of that processing, so you can come in at any point within that process, and will provide human coding of the major fiber tracks on the data sets for subject 1 to be public, and for the others we will use internally for scoring. The MRI data will be providing high resolution MP-RAGE structural images of the individuals. In terms of diffusion techniques, there are 2 leading techniques in the field at the moment for dealing with the fiber crossing. One of them is high angular resolution single shell. In that case, we look at diffusion in 256 directions and gives us an ability to plot the direction of the fibers and examine multiple fibers within that process. We thank Danny Alexander for giving us advice on how to optimize those scans that we will be doing. The second major class is the multi-shell methods. In these methods, one collects not one shell, but shells at different levels of diffusion extent, which this method, some groups feel, is better at resolving particularly triple crossings within that set. And we want to thank Patrick Hageman for advising us on how to optimize that pulse sequence. In terms of the MRI data for the structural and diffusion, we will collect MRI data for this on all subjects. We'll provide a resting state, so you can use resting state analysis methods in your segmentation element. We'll also provide for subject 1 and use internally for the other subjects the retinal field maps and auditory field maps, the motor system for the hand, foot, and tongue, the language systems, Broca's area and Wernicke's area, and the cognitive control net for attention, task decision making, and sequencing. We will also provide processed data. We will process through what we view to be some of the best techniques available to data and make the processed data available via those techniques. You can utilize that data either in raw form or the raw form data or the processed data. For example, we'll provide gray-white masks of the subcortical areas that are labeled. We'll provide free surfer 3D volume maps, the gray matter, the surface, the subcortical mappings. We'll provide diffusion-weighted deterministic and probabilistic tractography of the maps that are involved. And we intend to provide probabilistic tractography of that process. I note some of these analysis methods, for example, the last one, take enormous amounts of computer time, 10,000 hours to be specific. We are running those jobs at the moment, and we'll make those data sets, the analyzed data sets, available for the competition.